push on through it,
Okay,
deal with it.
good.
You know, I mean, I think that's pretty fair.
That's kind of weird for a teacher.
A teacher can care about her students.
Love and caring, you know.
But love is just a hair bit too far.
I think maybe you're thinking of love in a very limited.
All right, can we get on with this?
I'm just trying to explain what love is to Wyatt. Is that okay?
No, it isn't. Something that's.
It's Valentine's week,
Feel
Wyatt.
human emotions.
Okay. If I kill them, it's not on camera.
Oh, she's recording, I forgot.
Wait, can I put my finger right?
Free.
Control yourself today.
You won't.
You should flip the camera.
Braden wants us to move on. Oh boy, we are way behind. So, here is my DNA double helix. Please do not talk while I'm talking so I can explain it very clearly and not have to repeat myself a bunch of times. Hopefully you have this copied down. The first thing that has to happen with DNA replication is the DNA has to unzip and open up. So it breaks the hydrogen bonds. There's this cool little enzyme that lands right here, kind of this blobby little enzyme.
Kyle's mom's calling.
No, she's not.
I'm in the library.
His phone's not on.
That's not the phone.
My mom's calling.
So let's see, right here, this area is called the replication fork. It's where the DNA is opening, like a fork in the road. And this is helicase. This is a new word for us, helicase.
I saw it on the video.
Yay, science.
Did you?
I'm going to label a couple other parts while you guys are drawing that. The blue spine here is our sugar phosphate backbone. Make sure you're labeling all the same parts. Phosphate. And the little green guys are bases. I'm going to name those. Those. Braden, you probably know what goes down here if this is the complementary pair.
T.
Nice. Oh, of course. State markers. How is this marker dead? Hey.
Died.
I
Like
killed
how?
it.
No, I don't use them that much. Okay. I'm not going to fill out all of those. That takes an insane amount of time. I'm just making this up anyway. Okay? So, we have our sugar phosphate backbone. That is, it's going to apply later that it runs in opposite directions. Our bases are little green hash marks here. First thing that has to happen is that DNA helicase has to open up the DNA. Its job is to unzip it and break apart the hydrogen bonds. Okay? Helicase unzips DNA helix. Okay? The way I remember that pretty easily is the ending ASE means enzyme. And then helic is helicase, so it's an enzyme that breaks up a DNA helix. Okay? It never gets to come back together again. Very sad.
Maybe you just have to read your song about this.
Braden is in a mood today.
He is.
Braden Aragon.
Smoke.
Okay. So the DNA is unzipped, it's open, it's accessible. There is another enzyme that comes through and makes the new copy. Who remembers what it is?
Polymerase?
Thank you.
Nope.
Lovely. Let's draw it in blue. So we're going to start. Ooh, actually I want purple. Now here's, no, because everybody's talking all of a sudden. But. So the important part to remember is that from the video earlier, Benjamin remembers this because he wrote it down, is that they operate in opposite directions. So one strand is going this way and one strand is going this way. So I'm going to start with this strand. It's going to replicate continuously. It's also known as the leading strand. Okay. Leading strand. It needs one DNA polymerase. I'm going to draw it as this big blob on the chain here. Big boob. Okay. And brown. I'm going to label it as DNA polymerase so I don't forget later what on earth this big blue blob is. DNA polymerase. The definition you probably have in your book is that it joins individual nucleotides to produce a DNA molecule. More simply, ACE, the ending ASE, is an enzyme. Polymer or poly means many. So it's going to take single nucleotides and chain them all together to make a continuous strand. Considering that it's going to the right in this, it's already copied over here, so let's make that copy that it's already done. So green. Okay, so it's going this way, meaning that it's already copied this. So what letters do I have to add in here? We'll do them now.
A, T, T, C, G, G, C, A.
Perfect. Okay, so it's copied up to this point, and it gets to copy continuously. This one DNA polymerase is going to go all the way down the billions of base pairs and make a brand new double helix DNA molecule with one DNA polymerase. Just zoom along and get it done. Right, Wyatt?
I have a question.
Yeah, I mean, Emmanuel. That's what I'm actually looking at over there. He's looking shitty. What's your question?
So,
Hey,
What?
I'm recording. Please stop.
you put ATG, and then for the very top one, and then for the very bottom one, you put TAA, and then at the end they're both GCC. So you
I
can
got
switch.
baking soda.
Do tell me what your question is. Are you asking a question or are you just pointing out what I did?
Yeah.
I'm pointing out what you did wrong.
I didn't do anything
She
wrong.
didn't do anything wrong.
Well, because don't they have to be switched? The G has to be a C.
No.
They are.
There, so it's complementary base pairing, meaning that when they pair up, they have to pair up with A and T. A and T have to pair together, and
No,
C and G
look.
have to pair together.
Yeah, that's what I
Those are
thought
the
too.
same. Those are the same, but those are different.
Oh, I see what you
Yeah.
I see.
That's what I thought.
David.
I see. Thank you, Wyatt. I wasn't. You were just kind of saying this one, this one.
Dude,
I
you're the
wasn't
next
sure.
Einstein, dude.
But good catch. If anybody has that written wrong, better fix it. It should be C, G, G, C. Thank you, Wyatt, for cluely leading.
I'm a girl.
What? Close enough. Okay. Back to it, ladies and gentlemen.
I got
Listening, Jeremiah. Okay. So, our DNA polymerase, we have to have two DNA polymerase. The first one copies continuously down the leading strand. It's going to follow the DNA helicase and pull nucleotides from the cell and copy, or from the nucleus, and copy continuously and make a brand new double helix DNA strand to go off into a new cell and code for your eye color or whatever its job is. Okay? The other one operates a little different. I'm going to draw my arrows like this. It's going to replicate in chunks. This is what's known as the lagging strand on this side. This up, I'm really mad at you.
Did it mess up?
A little bit, you turd.
Who did that?
Who would do such a thing? Here we go. Okay.
I wonder who's banging the baking soda on the table.
You know, some people's kids, right, Wyatt?
Someone
Some
black.
people's too.
Okay. Now we have a DNA polymerase on here too. Draw that bad boy right here. Yeah,
Got tamales in
here
there.
he is. DNA polymerating right now. And I'm going to label it.
Got it.
Okay,
You got tamales in there?
Stay with me, people. Stay with me.
trying.
I know. I know. So, leading strand, DNA polymerase gets to just copy all the way down, just follow the DNA helix until it gets to the opposite end, and it's done. Because the sugar phosphate backbone goes in the opposite direction here, and DNA polymerase can only operate in that one direction, it's going to latch on at the helicase and then copy until it gets to the end. And it's going to hop off and go to wherever the DNA helix got to, or helicase got to, copy a little more. The DNA helicase is going to keep moving down, and it's going to do it in chunks. So if I'm drawing this correctly, this DNA polymerase is going this way. So it's going to have copied all of these. I'm not going to fill in any letters. We're just going to... Okay, it's copied to this point. What letter goes here?
T.
No letters.
Slow down.
Z.
Has the DNA polymerase gotten here yet?
Where did you get Z?
So nothing goes here yet.
Z.
The DNA polymerase is going this way, so it hasn't copied copied here yet, so I'm leaving this blank on purpose. It has not copied that far yet. Yes.
In the DNA polymerase
Pol-
is it never mind. It was
If it comes back to you, or I'm not sure.
I don't know. I was confused.
It's okay. So, that's basically summarized. The only other thing I'll add, if you're one of those honors test-taking kind of kids, there's a little chunk in here. When it's copying on the lagging strand, there's a little gap in each of these because it replicates back, and then back, and then back. There's a chunk right here missing. So we have something called Ligase in our cells to seal those chunks up so it's a complete strand. Like glue. Ligase means to ligate or link or glue together.
Also saw that on the video.
So now it's time to, I'm going to have you guys take 30 seconds to make sure you have this drawn right, to process and not get on your phones, and think of questions that you probably have.
I have no question.
What?
But you're still going to think on it for a minute. Just kind of stare at your picture or mine.
Just make it look like.
Think about it for a
Where
second.
is this located in our body?
In the nucleus, in every single cell. This is where it's happening.
Where's the cell located?
That's
Right there.
not-
Cells, trillions of cells everywhere.
Can I go to like
Hold on.
executive wall?
Can you and Sarah marry each other?
No, I asked
We're
her-
still processing right now.
I asked her.
After we process,
I asked her.
After you can ask a good question about this.
I asked her.
Okay. For including you guys. You're all.
What are the little blue lines?
Hunter has a question for you, Noah. What do you got?
Yes,
So,
Hunter.
Quiet,
where are
please.
the extra nucleotides hanging out? Are they just like float?
The nucleus makes them. It's actually the job of a ribosome to make those nucleotides. They're making like little sugar phosphate base chunks, and it just pulls them
I
out.
have the same question.
How does it know which base it has to pair with? Like, how did it know that it has to pair with T if it's A or C if it's a G?
soulmate.
I mean, it's chemically how it happens. Basically, it links up. It can happen the wrong way, and they won't pair correctly. There is this little.
What happens when that happens?
Well, there's actually a mechanism. I'm getting myself a little confused, but I can't talk over you guys. There's a mechanism, there's another enzyme that follows behind the DNA polymerase that proofreads it and double checks that it's been doing the job correctly. And if it finds like a C with an A, it'll delete it and correct it. Sometimes it gets skipped over, but there's billions of base pairs, so it works most of the time. Yes, sir.
How long does it take for this to happen?
Oh, very fast.
Like how fast?
I don't
30
have an
seconds?
exact answer for that because it's billions of base pairs, but it's a lot faster than I can draw it. Hey, Destiny. I'm just saying hey for the video.
Oh, hi video.
I can't say how long exactly because but it happens a lot faster than I can draw it. I can say that much. Benjamin Hall.
When the thing's done, when it goes to that loop, does it restart or does it just go one?